Hello and welcome back. We were just finishing up Gregor Mendel and his groundbreaking work in genetics last time. He ends up cracking the code. He figures out that there are two genetic factors. We didn't call them genes, he called them factors that contribute to a trait appearing in an organism, and that there's a dominant and a recessive. If the dominant is present, it will express itself. Figures out the genetic ratios and all that kind of stuff, which ends up getting simplified in a Punnett square eventually, but he didn't draw those. Those don't come till later. Now, news does not spread like wildfire. Unfortunately for Mendel, he is, so unfortunately / fortunately, he does eventually make it at being a teacher. He's loved by his students, and his favorite subject to teach, believe it or not, is physics. So his students just think that he's brilliant, and they learn a lot from him, to the point that his photograph was preserved at the school where he taught, and many other things that he had built and done, and he's remembered fondly there. However, his work with pea plants was misunderstood or misinterpreted, or I'm not really sure what happened, but they considered it another study on plant hybridization, which of course they had many of those studies. And they basically ignored Mendel's work in this area. And it wasn't until the early 1900s that it was realized that Mendel had cracked the genetic code. If I remember correctly, it was actually a bookworm, a librarian type person that was going back through some of the old archives and just reading old books, I guess, and stumbled across his work and had the intelligence to realize that this is more than just a study of hybridization of plants, that Mendel had figured out what was going on with genetic information. Now, at that time, 1920s, 1930s, evolution has been taught and practiced, and the whole degree fields have opened up in the teaching and understanding of evolution, attempt to understand evolution for decades. Entire generations of humans have lived and died teaching this hypothesis. And so it's very deeply ingrained into the psyche of Europe. But this is about the same time that evolution is starting to make inroads into the United States. United States, remember, when Darwin publishes his book in 1859, is on the brink of the bloodiest conflict we've ever fought in terms of percentages of humans that were killed or injured. The American Civil War, over 4 years of insane conflict, and then an attempt, you could argue, kind of a failed attempt at Reconstruction, abject poverty in many places in the South. You know, a lot of people try to rebuild afterwards, and it's a very slow process to get everything kind of back on its feet. And we got the westward expansion and the building of the railroads, and we got all kinds of better things to do. Actually, end up fighting some First Nations folks as well during that time period. So America was busy. We weren't super interested in what was going on with evolution. There's a much stronger resistance to it in America. And so it is in the 1920s and 30s that evolution is finally starting to get into our colleges and universities, and there's professors and folks like that that are starting to preach the evolutionary sermons, and trying to convince people that the earth is millions of years old and we all evolved from apes. And you've got the what's his name? I think Scopes, the monkey trials, and all those kinds of things that are happening. So right in the midst of that, you have a revolution in science, the discovery and study of genetics. We need to understand that this was a crisis for evolution. The realization that you cannot appreciably change the genetic information that you pass on to your children. Because, of course, Darwin had assumed that a horse out in the savannah stretching its neck out a little bit further and a little bit further will eventually evolve into a giraffe over perhaps dozens of generations, but it would evolve into a giraffe. We now know that's nonsense. Oh, by the way, there was a study on this, I may have already mentioned, involving mice. Mice, of course, have tails that are kind of strange. You know, if you read Narnia, the mouse has a sense of pride in his tail, right? But they don't really have any muscles in their tail, they don't have any nerve endings. They don't really use it for anything. So the hypothesis was, okay, well, if Darwin was right and things evolve by use and disuse, then removing the tails on mice should show some effect in their children. And so this scientist who wanted to demonstrate that Darwin was right got a bunch of mice, cut off all their tails with carving knives, whatever he was using. Sorry, a little bit of humor there, attempt at humor. So he noticed that all of the mice babies were born with tails and cut off their tails as soon as they were born, and did this for many, many generations, and never saw a reduction in tail size as a result of his efforts, because of course, we understand he's not changing the genetic information. And so, of course, the mice are going to be born with tails. Okay. So, realizing again, about that same time as evolution is trying to make progress in America, you already have some resistance. We're not seeing the results from Darwin's work that we should be seeing. We're starting to cast doubt on Archaeopteryx as a transitional form. There are doubts about Piltdown Man. Why are they keeping it locked in a vault? You know, all those kinds of things are happening. So in America, you have some folks, at least a few, that are starting to resist this philosophy and do the necessary work to fight against it. Okay, my spiritual great-great-grandfathers, I suppose. Anyway, the scientific community that is trying to push evolution is not excited about genetics, and they are not going to believe it unless they absolutely have to. They're, as I say, dragged into an understanding of genetics, kicking and screaming. And there are many of them that duplicate Mendel's work, trying to find an error with it. There's one gentleman in particular whose name I have not memorized because he is not worth remembering, that duplicated Mendel's results with pea plants. He got the same results as Mendel in an effort to disprove genetics. And when he published his work, he was like, look, I got the same results as Mendel, but that doesn't mean that genetics is true. He had gone and verified all of this scientific knowledge and still would not believe. Because, of course, in order to accept the simple truth, he would have to destroy his philosophy that everything had just built itself by random chance over millions of years as perfect nonsense. So, what is the evolutionist answer to an understanding of genetics? It is the second flavor of evolution that comes about around the same time period, evolution by mutation. It was an understanding that really the only way you can change a genetic code is by mutation. And of course, we understand mutations are almost always fatal and very rarely bad for you. And 1 in 1,000 or 10,000 might be benign, but there's really no such thing as a beneficial mutation to speak of. We'll talk about that in a couple of slides. Mendel, of course, you might have expected him to put this theory forward, and you know he's on the cutting edge. He knows he's discovered something very, very important. But this is a strange thing about humans, is we put so much stock in what other humans think of us. Even revolutionary humans, even geniuses like Mendel, still put a lot of stock in this kind of thing. So when Mendel gets a big yawn from the scientific community, he sort of also puts a stop to his work in this field. And as I say, turns his attention to teaching. He's distracted. He spends his life fighting against government control and overtaxation of his school and trying to stand up for religious freedom. Very, very interesting. I think he might have been right in putting his efforts towards that goal. Of course, we know he was ultimately semi-successful, right, as far as religious freedom and government control over schools are concerned. Okay. Okay, well, when this comes to light, it's a crisis, as we just talked about. The only way genetic code is going to change is by a mutation, and that became the evolutionary mechanism. There's no other choice than evolution by mutation. If you're going to change an organism, you have to change its genetic code, and the only way to change a genetic code is to mutate it, right? And so we got all the jokes about, well, if you throw enough rocks at a black and white TV, it'll mutate into a color TV. No, it won't. The whole jokes about, well, how many times does a tornado have to go through a trailer park in order to assemble a 747? Can't do it. You cannot take information and make it better by adding energy or corrupting your data on your computer. Never helps you, does it? So making random changes to gene code, almost always fatal for an organism. Okay, so but that is the second type of evolution. So you had the first type, which was Darwinism. Okay, Darwinism is evolution by use and disuse. That is the discarded hypothesis. Nobody believes that anymore. It's perfect and utter nonsense. The second flavor of evolution is neo-Darwinism, or the new Darwinism. That is the flavor that is taught in every government school in America as some kind of a biological fact. Evolution by mutation. Simply mutating your genes over a long enough period of time will create change in an organism that will be positive. Okay. But nobody actually puts that into practice. They may say they believe it, but they don't practice it. Nobody sticks their head in a microwave. Nobody is out there drinking poisons and, hanging around nuclear reactors unshielded and just trying to mutate their cells as much as they can in order to evolve. Okay. So important to realize the hypocrisy of modern science. A lot of people complain about the hypocrisy of Christians, and there are a lot of Christians that wear a mask. That's what Hippocrates means, I think, in Greek, means to wear a mask. Well, the scientists say they believe in evolution by mutation, but they all treat mutations as if they're bad, don't they? They avoid mutating themselves as much as possible, just like everyone else. We don't want to blast ourselves with X-rays and gamma rays and, drink toxic waste and expose ourselves to harmful mutations. That is harmful. It's harmful for us. It's deadly to our cells, and it certainly doesn't result in, something like the X-Men coming around and, Magneto and all that kind of stuff. That's just perfect nonsense. It's fairy tales. It's science fiction. It will never happen. It cannot be observed in a laboratory. So two different flavors of evolution, and both of them, in my opinion, are rather ridiculous from a scientific perspective. Now, there's an article here on the Institute for Creation Research website. By the way, that is a fantastic resource if you're interested in further study on almost any subject that we're covering. There's just tons of information out there. Some of it very technical, some of it very practical, about further study. And, do we see this kind of thing in nature? Can you mutate something and cause it to evolve? Well, let's have a look at this. And let me make sure I've got the right screen share. There we go. Looks like Zoom is following me around properly. Okay. So if you can mutate something and cause it to evolve, then you want to be able to test this on simple organisms, right? Like fruit flies. Lots of scientific research is done on fruit flies. They have a very high reproductive rate. I want to say it takes them 20 days or something like that to give rise to the next generation. Nobody cares if they die. They don't have souls. They're not cute, those kinds of things. So we use them quite a bit in genetic research. Okay. So 600 generations of fruit flies. If you're going to compare that to 600 generations of humans, this should be somewhere in the neighborhood of 12 12,000 years worth of human evolution. Again, I don't believe any such time ever existed, but that's just my opinion. I don't have a time machine to go and test that hypothesis. 12,000 years is a pretty long time. You ought to be able to see some kind of change in an organism over 600 years. Again, if evolution is true, we should be able to test that idea and see it sort of fast forwarded in a simple organism such as a fruit fly. However, we don't see that. You go out and mutate a fruit fly, and basically the only thing you're going to do is make a whole bunch of dead fruit flies. And here is the proof of that truth. Okay. Yeah, a lot of Americans, something like 50% or so, believe in evolution, the big picture story at least. So if we could actually document evolution in action, evolutionists could actually vindicate their worldview and actually cite real laboratory research to support their crazy ideas. In 1980, that is quite a while ago, this search for proof led researchers to painstakingly and purposefully mutate each core gene involved in fruit fly development. I think there's 20 or 21 of them, something like that. So they did quite a bit of work trying to get this thing to change into something else. The experiments proved that mutation of any of these genes, mutations that would be essential for it to evolve into something else, merely resulted in dead or deformed fruit flies. Well, what kind of fruit flies did they get? Well, they had fruit flies that had no eyes. Well, they got most of them died, right? They had fruit flies that had curled wings. They had fruit flies that had wrinkled wings. They had fruit flies with legs for antenna. They had fruit flies with really disshapen mandibles that couldn't feed properly. They had fruit flies with extra hair. They had fruit flies with almost no hair. They had fruit flies with red eyes, fruit flies with white eyes, fruit flies with all kinds of mutations, fruit flies with no wings, which I suppose you would have to call a land or a ground. I mean, it's not a fly anymore if it can't fly, right? So, yeah, no evolution for 600 generations. And you might argue, being the critically thinking person that you are, wait a minute, that's only about 12,000 years worth of, if you're going to argue for human evolution. So maybe we just need to keep looking. And of course, that's the answer of the scientists. The scientists doing this experiment, however, the guy in charge of the research group said, well, apparently the fruit flies have reached the limit of their evolution. They can't evolve anymore. Well, yeah. By the way, he spent 15 years of his life doing this. How many millions and billions of dollars of taxpayer money and research money has been wasted trying to answer questions that can't be answered like this? What kind of potential, what kind of brain power is going to waste over this nonsense? Quite a bit. Well, again, you might say, well, we should just keep looking. Well, Michigan State University evolutionary biologist Richard Linsky and his colleagues searched for signs of evolution in bacteria for 20 years, tracking 40,000 generations. In the end, the species that they started with was hobbled by accumulated mutations, and only changes that occurred were degenerative. By the way, you still have scientists running around trying to say that they have evolved the bacteria. Well, you see, they've got a bacteria that you see it's hobbled by accumulated mutations. They've got a bacteria that can only grow in that lab. It is specially adapted for that, and it does a pretty good job of it. But that's all it can do. It can't survive in any of the other situations that the regular old garden variety bacteria could survive in. Okay. So yeah, again, you get a lot of science media trying to say they evolved the bacteria. Well, what did Alan Linton have to say about this? Again, he's an evolutionist, so he should be able to see the emperor's clothes if the emperor is in fact wearing any. What does he have to say? Where is the experimental evidence? None exists in the literature claiming that one species has been shown to evolve into another. Bacteria, the simplest form of independent life, are ideal for this kind of study. Generation times of 20 to 30 minutes. And populations achieved after 18 hours. In other words, you get billions of them in less than a day. Wow. But throughout 150 years of the science of bacteriology, that is going back to Pasteur, right? There is no evidence that one species of bacteria has changed into another, in spite of the fact that populations have been exposed to potent chemical and physical mutagens, and that uniquely bacteria possess extrachromosomal transmissible plasmids. In other words, and you may not have been familiar with this, bacteria can actually exchange useful genes with each other, which is one reason why, for example, antibiotic resistance becomes such a big deal so fast, because the mere tiny fraction of 1% of bacteria that occur naturally that are antibiotic resistant can share that gene with other bacteria if they find themselves in the presence of an antibiotic. So that's why it's super important to finish your antibiotic medication. You know, you get it for however many days, and the doctor says, now make sure you actually finish this. You're going to feel better today and will feel a lot better tomorrow, but keep going with the antibiotics to make sure that all these bacteria are totally dead, right? Because you don't want to have a relapse. If you do, you'll relapse. And yeah, most of the bacteria that are now infecting you are resistant to broad spectrum antibiotics. That's a problem. We've seen a rise recently in America and throughout the developed world of people dying from infections. And that will probably continue to happen until we figure out how to properly manage antibiotics. I think many scientists and doctors agree that antibiotics are overprescribed. And yeah, a lot of humans really just don't take them as seriously as they should. So I don't know, education might be helpful, but then again, humans got to take responsibility for this kind of stuff as well. Right, so bacteria possess these kinds of things. So if they did have a positive mutation, they could spread it throughout the population. So you would think that a couple of dozen or a couple of hundred of those, and you'd have a whole different species of bacteria. Nope, never has happened. Interesting, at least not in a laboratory, right? Since there is no evidence for species changes between the simplest forms of unicellular life, that is monerans, right, prokaryotic bacteria, it is not surprising that there is no evidence for evolution from prokaryotic to eukaryotic cells, let alone throughout the whole array of higher multicellular organisms. Wow. Yeah, again, getting into details on this fruit fly study. Yeah, after 600 generations, yeah, you just don't get an appreciable amount of difference between these guys. Adaptation is not associated with classic sweeps. Newly arising unconditionally advantageous mutations becomes fixed. That's not the case. That's not the case. So we have tested this experimentally. And really the question in my mind and the question in everyone's mind is, how much evidence do you want? Now, you notice I said, how much evidence do you want, not how much do you need? I know how much evidence they need. They won't accept any evidence. So they can't be reasoned with. So I ask people, how much evidence do you want? Hopefully that causes them to think critically. Usually humans are incapable of critical thinking. But that causes them hopefully to critically think and say, wait a minute. He might have a point here. Maybe I should have some standard which, if met, would cause me to call these things into question. Maybe I should have some requirements for these things in my own life, in my own mind, to be scientifically verifiable before I build my whole life on that foundation, my whole worldview, my whole understanding of who I am and what I'm doing in the universe. This is a big deal. So maybe I should put my feelings aside and my training, and maybe people have been trained to believe in evolution their whole lives. Right, well, why? Why do you accept that as true? Shouldn't you test and make sure that it is? Well, so we don't see evolution in in the laboratory, that's for sure. All right, so what are genes anyway? Good question. Watson and Crick figured out the double helix structure of DNA way back in the 50s. Okay, before then, believe it or not, before then, it was actually known the structures of these molecules, adenine, thymines, cytosines, and guanines that make up the DNA. Okay. And these guys, if they're like this in the picture, but if you turn them like that, the A's and T's fit together and the G's and C's fit together. And if you keep stacking them up, they actually rotate a bit every time they're stacked up. And you do that for a couple of hundred base pairs and you get yourself the double helix of DNA. And again, that's what Watson and Crick discovered with their understanding of biochemistry. Okay, so that's kind of what the structure is. It doesn't really look like anything, but what the structure would look like if you could see it, but you can't. It's way too small to see. But again, we see the bonds between them are hydrogen bonds, which are pretty strong, but not nearly as strong as the covalent bonds that hold the molecule together. So fairly easy to keep these guys together because of the hydrogen bonding. Fairly easy for helicase to come down and separate them out and copy one side, because again, it's a hydrogen bond. You're not going to damage the rest of the structure. And once your copy has been made, the DNA will come right back together because of that hydrogen bonding pretty easily. Okay. A's, T's, G's, and C's make up a code for a protein. Proteins are made up of smaller molecules known as amino acids. These, arranged in just the right order and folded into the correct shape, are what make your cells different from dirt. You're made of the same stuff as dirt is. It's just the way you're put together is unfathomably complicated, okay? So the 3 letters in DNA code for each amino acid. Alanine is an amino acid. It is coded for GCC. By the way, some amino acids are coded for by more than one code. So that's kind of helpful. Who wrote this code? Who put this information in the cell? According to evolution, nobody did. It just wrote itself. The appearance is just looking like, it just looks like it was designed. So how possible is it exactly to generate a specific code by a random process? We can calculate this. Now, you may remember, hopefully we've talked about it quite a bit, that it's actually impossible to absolutely prove anything in science, right? We have laws in science, most of those are in physics, aren't they, about how the universe works. And we're pretty sure about a couple of things: the law of universal gravitation, the law of electromagnetism, the law of entropy, the law of mass conservation, and other things like that we're basically certain of. Okay, but we can never be completely certain, can we? There's always a chance that we've been looking at it the wrong way. So we kind of hold our science with an open hand. Even though, gravity, for example, is a universally recognized as, a law in the sciences. Everything with mass is attracted to everything else with mass throughout the entire universe. And we can see that the law of gravity applies just as much in Andromeda as it does in our own galaxy. It applies in other solar systems just as much as it does in our solar system. We can see it working in multiple star systems and, galaxy clusters and all that throughout the cosmos. Okay, so we have actually much more than mountains of evidence that is true, and yet we still don't treat it as some kind of an absolute truth necessarily. Okay, so there's really nothing that we can prove for absolute certain in science. But you can prove things in mathematics. Okay, mathematics has proofs in it. Interesting. So we're about to apply mathematics to the real world and try to figure out how difficult it is to build a simple system. So maybe a practical example will help us wrap our minds around all this math. So if you want to build a system, let's say it's a simple system, it has 3 parts. How many different ways do you have of arranging those in a line, right? Well, you can do a calculation with this. This is called a factorial calculation. That's what that little exclamation point means right there. 3 factorial, if you put it in your calculator, is 6. And how did the calculator do this problem? Well, it said all the whole numbers between 1 and that number. So 1 times 2 times 3 times, well, 3 is the number. So 1 times 2 times 3 is 6. There are 6 different ways of arranging these things in a line. Now, let's suppose you have 5 different colors of Lego. Now you have 5 factorial different ways of arranging them in a line. 1 times 2 times 3 times 4 times 5. Well, this number is growing kind of quickly, isn't it? Suppose you have 10 different colors and you want to arrange them in a line. 3.6 million possible combinations of 10 different pieces in a line. 50 different Legos. Well, again, they all have to be different. Not necessarily, I guess you could use different shaped Legos as well. There are 3 times 10 to the 64th power different ways of arranging a system of 50 different parts. Wow. A factorial calculation, by the way, grows faster than any function in mathematics short of a double exponent. 100 different pieces, almost 10 to the 158th ways of arranging a system of 100 different Legos. You don't have to take my word for it. You can just go punch it in a calculator. Okay, so if we look at a calculator real quick, right over here, I've noted that this is a scientific calculator. Most computers will make sure I'm sharing the right screen. Yeah. So most computers will show, if you click on the options here, you can have a standard or a scientific or a graphing, and it's just a little fancy. So you want a scientific calculator, and you can put in 5 and this little N to the exclamation, or sometimes it'll be an X exclamation. That is factorial. Okay. And hopefully my computer is not going to crash on me, but 100 factorial. Yeah, there you go. By the way, careful trying to do very large numbers like 200 or 300 factorial. You might end up crashing your calculator and your computer on that. So just a word of the wise. You can do this on your own. It's 1 times 2 times 3 times 4 times 5, all the way up to whatever number you're trying to calculate. But I want you to understand, okay, I want you to understand how big these numbers are, okay? Well, you can't. Sorry. Oh, I know what you're thinking. Okay, Cartwright. Seriously? I can't understand this? But I got a pretty good education. I got a pretty good head on my shoulders. Lay it on me. I can't. It's just a fact. These numbers are too big to understand. There's theories out there that really large numbers like this might be impossible for us to understand because they outnumber our brain cells. So when you see a number like 10 to the 80th, 10 with 80 zeros behind it, your brain can't comprehend it. It just says, oh yeah, it's a really big number. But how big is it really? I can't explain it. I'll tell you what. I'll try to explain how impossible it is for me to explain it. I might be able to do that. Okay, we're talking about atoms. Atoms are very, very small. A hydrogen atom is something like 10,000 times smaller than a visible light wave. Dude. Okay, so atoms are very small. Can we put that into perspective? Well, let's suppose we had a speck of dust. I'm in a pretty clean church here, so maybe I can't find one. Maybe I can find one. A speck of dust somewhere. Ah, here we go. I have a speck of dust on my finger. We'll call him Bob. Bob is pretty small. But if he was an average size speck of dust, there would be approximately the same number of atoms in Bob as there would be Bobs to make up the earth. In other words, a speck of dust is halfway between the size of an atom and the size of the earth. So you're probably aware of the fact that it's impossible for you to understand how many specks of dust it would take to fill this room. That's just an unfathomably large number. You can't even fathom how many grains of rice there are in a sack of rice. It's huge. Okay. Well, how many grains of or specks of dust would it take to cover the planet 100 feet deep? That's a lot. That's a lot of dust. And to make the entire planet, remember the Earth's crust is on average something like 20 miles thick. It only makes up 1% of the mass of the Earth. So how many specks of dust is that? So you understand how many levels of impossible it is to understand how many atoms there are in the Earth. But are we to 10 to the 80th yet? No. Okay. Working our way up. There's 318 times mass of the Earth in the planet Jupiter, give or take a teaspoon, right? Jupiter is almost twice the mass of all the other major planets put together. And yet, all of that mass put together, what would that be? That's about 725 Earths. That's less than 1% the mass of the sun. Wow. Okay, so how many atoms are there in the solar system? There's a few. Okay, and then you have to remember, we multiply our solar system by roughly 100 billion to get the number of stars in our galaxy. And then we have to multiply that number of atoms by roughly 100 billion to get all of the atoms in the observable universe. And you can do the math on this. I actually have and verified it. It is 10 to the 80th. That is all of the atoms in the observable universe. So is it possible to get a specific sequence in a system of any complexity, like the system of 100 pieces or the 200 pieces of a human skeleton? No. And by the way, you're not just aligning them up in a particular order. Human skeleton has to be arranged a little bit more complex than that. So I want to emphasize that these kinds of calculations are fairly easy to do. Okay. For example, you can actually calculate ribonuclease. Remember, we were talking about that earlier, I think last week. Ribonuclease is the simplest protein known to science. 17 of the 20 possible amino acids. So the chance of getting one correct, if you could in fact just poof, make amino acids, but you can't. But if you could, 1 in 17, that's not bad. That's probably happened in the entire history of the universe. But getting 2 in a row correct are 17 times 17. This is a type of factorial calculation. 17 times 17 times 17 times 17. And there are 124 amino acids in that protein. Again, this is the smallest one. A lot of proteins have hundreds of amino acids. Okay. And some of them, like ribosomes, are assembled in 2 huge pieces because they're so huge. All right. So getting all of the amino acids correct out to 124. And again, you don't have to take my word for it. You can do this calculation. It only take about 5 minutes. Put in your calculator 17 times 17 times 17 times 17 times 17 times 17, 124 times. You get one chance in 10 to the 152nd power of getting the smallest protein known to science correct. And I want to point out to you that 10 to the 80th, you might be tempted to think, well, yeah, this 10 to the 152nd, that's twice the size. No. These are exponential numbers. 10 to the 81st is 10 times the size of 10 to the 80th. 10 to the 90th is 10 billion times 10 to the 80th. So is it impossible to get a protein by random chance? Even if you had all the cellular equipment there to put the thing together, you cannot build a functional protein by random chance, even the simplest one. It is so far beyond impossible. It's almost like God is trying to make this painfully obvious that every single protein in your body was built by him. We bear his fingerprint. And we can be sure that we bear his image on our souls because of the evidence in the physical reality. Because we can see the fingerprint of God on our physical being, we can trust that he will preserve the spiritual imprint on our souls that we call the image of God. Now, that ought to be encouraging for you. And it's a little bit terrifying as well, because when I start thinking about all of the tens of thousands, hundreds of thousands of proteins in every single one of my 60 trillion cells, I start to understand how powerful this God is. It's not just that God exists. That's easy to prove. It is who God is that ought to freak you out. It is the power of God that should cause us to pause and reflect when we think about rebelling against this guy. We're not talking about a lollygagging, tiptoe through the tulips kind of a dude. This God holds your molecules together. Maybe we ought to find out what he wants. Maybe, just maybe, when God gives us instructions on how to live our lives and how to treat our neighbors and how to spend our money and what kind of clothing we should wear and what we should do with our time, who we should be hanging out with, maybe we should take this guy seriously. Just throwing that out there. Again, we're talking about, the smallest protein. There's lots of proteins out there way bigger than ribonuclease. Here is an X-ray crystallographic image of the structure of a ribosome. We have the active region here labeled green, the southern hemisphere, as it's called, just because it's a little bit smaller, labeled purple, and the northern hemisphere labeled blue. But of course, these represent a whole bunch of oxygens, carbons, nitrogens, hydrogens, smattering of other elements, all arranged into amino acids that are then arranged into a long protein chain. By the way, they have to be folded into this exact shape. What are the chances of folding it properly? Well, that's just the thing. We've figured out the chances of assembling the thing in a long chain. We cannot even calculate the difficulty of folding it into the correct shape. We know what happens in the Golgi body. We know the Golgi body modifies proteins. So you have the genes for about 25,000 different proteins, but your Golgi body may be able to make, we don't even know, maybe hundreds of thousands of different kinds of proteins from the basic type. And how does it know anything anyway? It doesn't have a brain. For heaven's sake, it's an organelle inside of a cell. But somehow it knows how to modify, not just to fold them in the proper shape, but tweak them a little bit to make the vast array of varieties of proteins that you have running around in your body right now, working tirelessly to keep you from turning into dirt. This kind of thing makes my socks roll up and down. What do they say these days? What do the cool kids say? Oh, that's the fizzle. Is that, do they still say that? I don't think they do. We're bringing it back, the fizzle, okay? Well, if this image doesn't impress you, just keep staring at it for a while. Doesn't it glorious? This is one of my favorite scientific images because it's kind of like staring at the moons of Jupiter through a decent telescope. If you're anything like me, hopefully you're not, but if you're like me, you need humbling experiences in your life, and you sort of get this idea that you got it all under control and you got it all handled. And hey, my classes are going pretty well, and my relationship with my family is going pretty well, and everything is just kind of working out. And I'm kind of an awesome person. Yeah, if you're anything like that, I need this kind of reminder in my life on a regular basis that I'm answerable to the guy who designed this. I got to give an account to him of how I've lived my life. And honestly, that's going to be the worst day of my life, because there's so much that I've wasted, so much wasted effort, so many empty words, so many hurtful words that I've said, so many things I've done, so much time that I've wasted kicking against the goads and, trying to figure my own life out rather than following God's plan. Isn't that ridiculous? This kind of thing helps me be patient with my students. Not you guys are awesome, but some of my students are a little crazy. Well, if the guy who built this is still patient with me, maybe I can be patient with my fourth graders. Maybe I can try to be the teacher that they need. Well, we can actually animate this process. Now, I'm not going to take the time in this video to go and watch these videos and put them in the Zoom video that I'm making, because YouTube doesn't like that. In their minds, it is taking views away from the original creator of those videos. So, and that makes sense. So these links will be in the PowerPoint that I'll email out. So feel free to go look at them. The first one is the protein synthesis animation. And the second one is an animation of all of the steps involved in leukocyte extravagation, which is a fancy way of saying that's how your white blood cells get out into infected tissue, right? Because your white blood cells normally, if there's no infection, will just roll along the edge of your blood vessels, right? So your blood vessels are lined with a Velcro-like protein sticking up, and your leukocyte comes along, your white blood cell comes along and sort of shakes hands with them, and that's how it stays attached to the blood vessel wall. And it sort of rolls around along that wall of your blood vessel using the push from your blood pressure to push it along. And so it's constantly shaking hands and unshaking hands, and it's sort of doing this Velcro thing with the proteins on its surface and the proteins on the surface of your blood vessel wall. If you get an infection, however, there's what we call a protein cascade. There's a reaction that happens in your cell, and it freaks out and makes a different kind of Velcro, for lack. There's a specific name for it. It's in the video. You can watch it. He has a special name for it. Anyway, instead of shaking hands with the leukocyte as it's rolling along, this protein, and by the way, there's thousands of them that stick up like that. So it's not just the one or two. This one locks onto the leukocyte and anchors it in place and sends another signal into the leukocyte. The leukocyte dissolves its cellular structure and becomes flat, for lack of a better phrase, and squeezes out through the cells that make up the blood vessel wall and out into your body tissues to go and fight the infection. I show you this video. First of all, I think it's an extremely interesting area in science, and one of those things it's quite a bit beyond my ability to understand, but also to show you what's involved if we don't simplify our biology. This is the only video I show to my biology students that is not a simplified version of something. That's all the steps. Similar to blood clotting, except blood clotting actually involves 15 different steps in a protein cascade, similar to this one. This one doesn't have that many steps in leukocyte extravasation. So you'll see in the video, cellular structure dissolving itself. You'll see it rebuilding cellular structure. You will see in the video synthesis of new proteins and them being transported up the cellular highway by a little motor protein. That's the cutest little thing ever. But there's a little motor protein that literally walks up the cellular highways to deposit this giant container of new proteins that can actually grab onto the leukocytes. Say, help, we need some help over here. We got an infection. Part of your immune system. It's just unbelievable what's going on in all of your cells. Okay. And there are some who insist that it built itself over millions of years. Well, unfortunately for them, once you get beyond the cell, it doesn't get any better for you. So we'll talk about irreducible complexity in the next video. See you then.